It's my great pleasure for me to have a talk in this conference, and so I would like to express my appreciation to all organizers for such opportunity. So the title of my talk is an alignment model for orientational aggregations of interacting bundles of cells. This is a joint work with Passam, Angela Stevenson, Juan Velasquez. This work was motivated, during the time when I was in Max Planck Institute. At that time, the Angela Stevens was my mentor. Anyway, so my talk is organized as follows. First of all, I'll introduce the model and our considerations, and I'll speak a little bit about biological motivations and review some preliminary, maybe previous results. And we then state our main result, which are twofold. The first case is cells are attractive. And so basically the cells, the thing we are considering is the biological cells. So the cells interact each other, and one case is they are attractive case. So depending on initial distributions and optimal reorientation, which will be specified later, so that sometimes it develops one peak. Yes, this means one peak is one alignment. Every cell is aligned in one direction. And the other case, it could develop several peaks, several alignments, depending on, again, initial distributions and structure of the reorientational alignment. So the other case, the second case is we consider the case the reorientation or optimal orientation, which is sometimes attractive, sometimes repulsive. More precisely, if the cells are close, in terms of angles, then they are attractive. The angle is a bit far away, then they are repulsive. In such case, we are study some solutions which are symmetric case and also non-symmetric case. Of course, non-symmetric case is much more complicated than the symmetric case, but when it comes to computation, but in principle, the argument we use is similar. So in this talk, I just brief idea of how the proof goes for only the case symmetric solutions. Okay. So. Yeah. So let me begin with these previous results. Yesterday, Dr. Huang introduced the model derived by Othman Dumba-Alt, so I just skipped the derivation, but just to mention it in the result. So Othman Dumba-Alt derived the following kinetic model from a stochastic jump process, and also the Mogilla and Edelstein Keshet introduced various models of actin filament modeling the alignment of the biological cells. So what they did is they produced a lot of different types of models, and they linearized the equation. The equation is written basically in terms of a convolution, which has a non-local effect. But anyway, what they did is they just linearized near the stable solution, which means just uniform distributions of the cells, and they studied some stability problems. The model under our motivated our consideration is due to the study done by Gigant and Stoll. They studied some integral differential equations, which shows some alignment models of the biological cells. But this is a little bit simplified models in the sense that they only take into account the dependence of the angular dependence. I mean, they didn't consider the dependence on the spatial positions of the cells. So it's a little bit simplified models. So basically, cells interrupt each other depending only on the angle differences. Okay? And what they did is somehow they studied these models, and then basically bifurcation analysis was made, and also numerically they show that their study is valid. That's what it did. So basically our goal in these studies, we want analyze the model more rigorous manner so that we prove rigorously the peak is developed depending on what types of initial configurations and also what kind of structures are related to the reorientational optimal reorientation. Of course, I didn't introduce yet what optimal reorientation is, but we'll see soon. Okay. Unfortunately, my biological knowledge, the knowledge on biology is quite limited. So, but just in case you are interested in what the actin filament is, let me just give you, show two pictures. Okay. I just copied these figures. How can I expand this? Okay, so I copied these figures in Wikipedia. This is basically a dyed cell, and the blue is the nuclei. And the actin filament is the kind of a long rod, so it has a head and tail. Okay, and the size is more or less, as far as I remember, 7 nanometers, quite small. And basically, you can observe those guys inside of the cell. Mostly, everywhere you can find this inside the cell, but most of the actin filaments are observed near the cell membrane. And there, the actin filament is somehow aligned. So, of course, it randomly distributed in some regions, but if they are densely packed, then somehow they are aligned for one directions. So here in these figures, the actin filament is one of the major component of the cytoskeleton, and they participate in some cell motility and cell divisions or maintenance of the cell junctions and the shapes of the cell and so on and so on. But anyway, in these figures, you see the things colored in red, they basically actin filament. And also there is some green, which is another filament, which is larger than actin filament, which is microtubules, I think. Okay. So. Next one. Yeah. Sorry about this. Everything is written in Korean, so I just specify what actin filament here. Okay. So you see there is a small long rod here, right, near the cell membrane. Okay. So these guys are actin filaments, and near the membranes, they are aligned somehow. Okay. If you look at some local region near the membrane, the actins are aligned. Yeah. Okay. So next, let me just introduce the model studied by the Kyungkeun. So F indicates the cell density defined on the unit circle, because we are just considering the dependence of the angles, not the positions. So, our geometry is 2 dimensions, but it could be reduced to one variable function because it only depends on angles, right? Of course, the unit circles could be denormalized in the unit intervals. Of course, cell density is positive, and the model under our consideration is the integral differential equations, where the TF typically referred as the turning rate, which is again defined in terms, as an integral. Okay. So the meanings of the right-hand side is as follows. The first guy is the guy jumping from U to V, so it's loss terms. The other terms is a kind of gain terms, which is jumping from the direction U from the V to U. Okay. So here H is interaction rate, which is strictly positive, symmetric with respect to 0. But in our analysis, for simplicity, we assume to be 1, constant. And, oops. Okay, let me go back. Okay. G sigma is basically the Gaussian, periodic Gaussians. And MW is optimal reorientation, which I will explain later. Okay. And our analysis, as you know, if the sigma is getting smaller, then the Gaussian is narrow. It's getting narrow, which means the reorientation happens in a low variance. Right? In our analysis, actually, we consider the extreme case, limiting case, namely the Gaussian is Dirac mass, which gives deterministic turnings in our analysis. Okay? So, let me explain what the optimal reorientation is. So the measure of functions MW is reorientational, optimal reorientation, which is these things indicate somehow the interaction with, somehow the jumping angles due to interaction to the other cells, basically. So it might be better to look at these figures, but anyway, let me first show the reasonable form of the optimal reorientations. Okay. Keep in mind the system should be invariant on the rotations, so we could say the optimal reorientation might be of this form, V plus capital V, and this is the difference of the angles, where V is another function which is said to be a orientation angle. So, okay, situation is as follows. We have two cells, for instance, and then these guys interact each other, self-interacting. So this measures somehow change the angle due to interaction to W. Okay, V change the directions, right, by W. How? How much? By these angles, right? So MWV is the new directions of the cell, right, caused by the influence of the W, right? So it must be V plus some angles, right? Of course, the change in the angle must be depends on the difference between two angles, right? Okay, so that's the reasonable assumption, I think. So one of the easiest case is, of course, this is attracting case. If it is repulsive, then the angle is the other way around. Okay. One of the typical example is the V is like half times this angle. Then actually this is located exactly the half of this angle, right? Of course, you see, oops. If G sigma is not zero, then it's probabilistic. But in our analysis, because we are studied the limiting case, so the change in the angle is exact, right? Okay. So as I mentioned earlier, the interaction rate is just a constant functions for simplicity. Of course, we could do for under the reasonable assumption. Oops, I'm not familiar with this. Okay. So H is positive bound, but I mean, when it comes to the computation, it doesn't change very much. So for simplicity, we assume H is 1. Okay. And so again, our analysis is the case, the limiting case, namely the Gaussian is a Dirac mass. Okay? And then our main goal is, again, as I told you before, we want to understand the structure of the, the development, the peak. I mean, here is not aggregation. Somehow aggregation means the aligned direction. Every cell is aligned to one direction, sometimes two directions, multi-directions, or depending on initial configurations and the structure of the optimal reorientation, which I introduced before. Okay? Okay. Yep. Let me go down. Okay. So now it's time to specify the optimal reorientation is. Okay, so let me just remind you what optimal reorientation is, which is of the form, right? Okay. So, I mean, so all you have to do now, we specify the orientation angle, right? So the orientation angle should be an odd function with respect to 0, right? And it should be one periodic, right? That's the basic requirement, I mean, right? These are trivial things. And also with some restriction, like this is bounded by some linear functions at the endpoints. And then the linear function is not so steep, because A and B is a kind of a technical assumption, but I think it's reasonable in a way. So A and B are less than 1, which is also useful in our analysis. Okay, so here is somehow the typical types of optimal orientation, more precisely orientation angles, right? So this means the cells are attractive, the first case, okay? Attractive, and then the range is quite large. I mean, long range attractions. Here, the attraction is quite short. I mean, cells are attractive, but only the case the angles, difference of the angle is not so large. Okay? Then they are attractive. Otherwise, there is no response between cells. Okay? It's the case. And then the last case is, I think it's a more interesting case, so that if the angle is small, they are attractive. Otherwise, then they are repulsive. For instance, one of the cases, as I told you before, we studied the symmetric solutions, where the optimal orientation is also the symmetric. So if the angle is less than phi/2, then they are attractive. Bigger than phi/2, then they are repulsive. Okay, that's the last case. Yeah.
Earlier you said you have a picture of the cell, and there's a tube. Is this tube the sensor, you think?
I think the modeling is motivated by the biological things, but in a way it's simplified. So we just test only the cells, and then we assume they are self-interacting. There is no other things are involved.
No, I understand.
Yeah.
But you did mention the cell.
Yeah.
There's a tube near the surface.
Yeah.
Do you mean that this tube is a sensor to feel the existence of the neighbor or not? No.
I mean,
that tube, that's what made me think about why you made that tube.
Yes, I told you.
There's a filament, isn't it?
Yeah, it is a filament.
It's the backbone of the cell.
Oh, filament. Oh, okay. So it has nothing to do with the
Sensor.
Oh, not the sensor. Okay.
Yeah, it's just a filament, but okay, maybe I misunderstood your question, but the filament sometimes attached to the cell membrane. That's your maybe question. I don't know. I mean, as I told you before, my knowledge is quite limited, and this model is not only the cell filament, but there is a lot of different kind of. Okay, for instance, there are another examples which have some alignment, even if it is not. Filament. So I think our analysis is mostly focused on the kind of mathematical analysis, not okay. Okay, so good. Okay, so this is kind of a summary which I already explained before. So let me just explain again. The unidirection means the orientation angle is always positive, and because it's a long-range interaction, so it is strictly positive everywhere except for the endpoints. Okay. The multi-direction case is a short-range interaction, so the orientation angle is just positive within the small interval. Otherwise, it's just vanishing. It just vanishes. And also, I mean, I have to mention that not only this structure of the orientation angle, but also the initial distribution is important. So that, I mean, this is somehow the easy case, I think, I mean, to prove the multiple peaks. So if the initial distribution is already separated, then there is no interaction further, right? Because of the short-range interaction in this case. Then the behavior of the cells are somehow separated. There is no interactions as times go by. So it's like an individual problem, like one peak problems. Okay, then we expect several peaks. And in our analysis, is the most interesting case is the last case, bidirections. So our setting is as follows. As I told you before, orientation angle is positive for some angle. I mean, symmetric case, theta 0 must be 5/2. I mean, in our case, quarter, I think. Okay, I mean, in the normalized intervals. But after that, after the angle theta 0, they are repulsive, which means the orientation angle is now negative signs. And then the thing we study is the case, the initial distribution. Of course, they are separated, then it's treated as the case of the, attractive case, I mean, multiple peak. But the thing we have in mind is the initial distributions have some, almost separate, but in a way they are connected, not separated. Then, of course, you expect a similar situation. Eventually, the two peaks occurs, I mean, two different alignment occurs at the opposite side. But the analysis is not so easy because, in a way, if they are connected, then we have to take into account the directions, the middle guys, too, right? Okay, you'll see that later. So, but yep. Yep. So we are focused on the last case in our analysis. Okay. So let me briefly mention the basic stuff. Global existence of solution is not so difficult. Basically, our equation is kind of ODEs. I mean, even if the right-hand side has some integrations, and the way to prove is a standard. I mean, the Gronor types estimate gives global existence of solution as long as the initial data is in the nice space. Okay. Another assumption should be made on the initial distribution. For instance, if you want several peaks, I mean several alignments, then also, of course, you could think a more general situation where the initial conditions are not separated. But I think it's a much more difficult problems. Okay, so this is a simple situation. The initial distributions are separated, and so in case optimal reorientations as a short-range interaction, then develop several peaks. So that's the case of attractive alignment in our considerations. Okay. Okay. So let me just simply show how the proof goes for the attractive case. For instance, I mean, just the mainstream, not the computation. Okay. So unidirectional alignment is, so we have initial distributions. Everything is located within the angle 5/2, for instance. And the optimal reorientation is some attractive case. It's one sign, right? Positive sign everywhere, and then long range. Then, what I expect is, you immediately prove that the mass is conserved. I mean, from the equation, also the first moment is preserved for all t's. Then you could define the center mass, right? This is a time independent. And then the next thing, it's a typical thing, I think, okay? Typical thing we could do. So we just compute second moments near the QC. Then what I can show is actually this guy is decreasing as times go by. This is strictly negative unless F is exactly the mass times the Dirac mass at the position, center mass. Okay, so eventually what I expect is just everything's aligned in one direction. Okay, yeah, I think this is not so difficult. Okay, let's see. Okay, multi-direction is similar. I mean, in principle, it's like a unidirectional case because you see, as I told you before, we consider the case initial distributions are somehow separated, and then the optimal reorientations are short-range interactions so that everything goes well, I mean, separately. So, okay, but of course there is some technical difficulty, but I think in principle the computation goes through like one unidirectional case. Okay, so the way to prove is we show that the conservation mass for each regions, separated regions, and also we need to show the first moment is also preserved. Okay. Oops. Okay, then individually you could define the central mass, and then second moment is decreasing and so on. This is the same thing. Okay, so in principle everything is like independent island. So everything is treated as like a unidirectional case. Of course, eventually,
Excuse me, can
Yep.
you see the previous page?
This one?
I see. Okay, AI.
Yeah.
What's the AI?
Yeah, it's a separate regions. So initially, the initial conditions, there are many guys which are compactly supported, which are separated. Okay. Okay. Yeah. This is a simple situation.
Yeah.
Okay. Okay. More interesting case is the last case, attractive and repulsive case. So as I mentioned before, we study two types of solutions, symmetric case and non-symmetric case. Of course, you may think it doesn't need to be considered the symmetric case because non-symmetry is more general. But I think the reason we study the symmetric case is because at the beginning, the non-symmetric case is we didn't have any idea how to treat. So the starting point is we study the simple situation where the solution is symmetry. So which means somehow we know that already at the beginning where the peak develops and so on and so on. As long as the initial distribution properly assigned. Okay. Okay. So we need some assumptions. Not only the initial distribution, but also the conditions, reorientation angles. So orientation angles. We make this kind of assumptions. Again, I think this basically means some part is positive, some part is negative. Okay, and at the origin, it is. We already make some assumption like bounded by the linear functions, but also the strictly positive, and near the when the signs are changing, then near the point that the derivative is also strictly negative. That's a reasonable assumption, I think. Okay. So what is? Oops. Go down. Yes. Okay. The typical shapes of the orientation angle given like this. It's a symmetry with respect to quarter. Okay. And then bounded by these straight lines. Okay. That's what it made for the orientation angles. And we also make some assumptions, so initial distributions. at the beginning we thought that kind of general types of initial distribution, but it's not easy to prove. So some restrictions of initial distribution is as follows. So basically, the most of the mass is concentrated at the beginning in quarter and negative quarters. Okay, and that's what it means. And it's not separated, but the other regions here and here is quite small. It's not zero, but quite small. Okay, I mean, you may think intuitively, this is an easy case, but it's not when it comes to computation, because we have to. I mean, these guys are repulsive somehow because the angle is quite large. However, so you may think every guy is attracted to this and this, so eventually you develop a peak, but we have to take into account attractions near these guys too. So it will be computations messy because we have to compute such kind of influence too. Okay. Okay, this is the case. Okay. So our main result for the symmetric case is as follows. So again, because the initial distribution already looks like a peak, right, at the point quarter and the negative quarter. So eventually we need to consider the second moment near the quarter and negative quarter. And then we show that actually this guy has exponential decay as times go by, and also the mass at the point of quarter and negative quarter eventually is half of the original total mass. Okay, so which means everything is somehow aligned in one direction, and the other part, half of the cells align the other part, and then the difference is exactly 5/2. I mean, the development of the peak occurs at the other side. Okay, that's the thing. Okay, so let me just show how the proof goes. I mean, I just skipped all the details. Okay, so the way to prove it, let me just go back to the figures here. Okay, this is kind of initial distribution. Every guy is somehow concentrated near here, and the other part is not zero but quite small. Okay, that's the assumption. And when it comes to computation, not only these areas denoted by I delta plus, I delta minus, but also we separated, we introduced another region like A1 and then A1R, which is reflected regions, and this the other parts. Okay? And then, okay, so A1 and A2 are defined this way, and then we consider second moment and then supremum the other regions denoted by J. Okay, so what we want to do is somehow these guys, we want to see the dynamics of these guys and these guys as times go by. Okay, and then we compute. Okay, because we have to separate, treat separate many terms because one part is highly packed and the other is quite small, so that's why we have many terms. But eventually, for the time derivative of the second moment, eventually we have control in this manners. So you see, this is leading terms, but A is less. This is the assumption we made already. So A is less than 1. So it's smaller than these numbers. And the other part is just, J, the error terms, I would say. And also, we compute the other part, J of T. Okay? So, let me just skip the details. And then, what you have is the time derivative of J is the leading term has a minus sign and then quadratic T. So, if summing up, we have this, the system of differential equations. So you see the linear part has a negative sign, and the other is some error terms. So if T initially, we already made assumptions initially, so J and S is quite small, right? So then this OD gives some exponential decay as times go by. That's the proof goes, okay? All right. So now symmetric case is, again, it's a similar, I just skipped the proof and then I just state our main result, okay? So again, we make some assumptions on the orientation angles. Here, I mean, it doesn't need to be symmetric because we consider the non-symmetric case, right? And so the initial conditions, you see, when it comes to the initial distributions, we don't know, I mean, eventually where the peaks are developed. That's the one of the difficulties. Symmetric case, if the initial data is properly assigned, you expect the peak is already, must be there, right? But the non-symmetric case, it's not easy, right? So the thing we have to look at is somehow everything is more or less the same situation. Okay, let me show it. So the initial distributions are somehow concentrated near here, and then again this is quite small, but solution, I mean the initial data is not symmetric, okay? And also the optimal orientation angle is like that. Some part is attractive, negative, but doesn't need to be symmetric, okay? Okay. And then initial data, we assume that this means basically concentrate the opposite side, and then this is probably possible positions where the peak develops, okay? But it's not the case, of course. And this is a reasonable assumption, and so on and so on. So our main result in this case, we introduce a lot of different things. For instance, this is mass concentrated in this and this M2, and this is again the supreme the other side, and this somehow I mean, we trace actually the location where the peak developed in the end, okay? So also a second moment. So our main result is there is M1 tilde, M2 tilde, and then QC1 tilde and QC2 tilde, so that everything's converged to some numbers, so that the cells converge to, the M1 times the Young mass, C1, and then C2, and this should be total mass, I mean moment, and the difference of the angle, QC1 and QC2, exactly half. So that's the reasonable thing. I mean, that's expectable because you see, we don't know exactly where it is, but these guys must be within I delta plus, I delta minus, maybe not quarters, but near quarters, right? Negative quarters. But the the difference of maybe slightly change the position where the peak developed, but the difference of the angle is exactly half. I mean, half. Okay? Okay, that's what I proved. So let me make some conclusions and some remarks. Okay, this is the last slide of my talk. So, I mean, we study on integral differential equations modeling angular alignment of the self-interacting cells, right? And we proved that somehow I think we characterize the relation between initial data and then structure of the reorientational angle, so that if the situation is like this, then we expect that somehow development, peak alignment of the cells, and so on and so on. That's what it did. And recently, Primi, Stevens, and Velasquez consider the case. You do remember our case is the limiting case. I mean, sigma is equal to 0. But if sigma is not 0, then it's more probabilistic. So they approximate and models into kind of diffusion equations. It's approximate the equation of the integral differential equation in case the sigma is not 0. And then they show that some steady existence steady solution, and then there is a stability near the steady solutions, okay, by numerical simulations. Okay, so the final comment which I would like to is our model has some limitation in a way because only angle dependence, but in general it could be depending on positions or density of the cell and so on. So it could be more interesting questions. Okay, thank you for your attention.
Do you have questions or comments?
Is there a model